Hello and welcome to this webinar brought to you by the SATB2 Gene Foundation. I'm joined today by Dr. Jenniffer Fish of the University of Massachusetts Lowell, who's a key researcher in the field of SATB2. Dr. Fish is going to walk us through the different types of mutations and how the consequences of these mutations could result in SATB2 associated syndrome.
Hello, my name is Jenniffer Fish, and today I'm going to talk to you briefly about the types of genetic mutations and their consequences. And the goal of this presentation is to help individuals of the SATB2 Foundation, individuals, patients, and their families better understand how the specific mutation they have may contribute to the disease. So to begin with, I just want to provide some basic background on the relationship between genes and proteins, or DNA and protein. And that's important because it is the protein that is doing the job, if you will, that is performing the functions within the cell that are important for the way that cell functions and the way a tissue functions. So as many of you may be aware, what we refer to as the central dogma of biology is this relationship between DNA, RNA, and protein. So DNA is the genetic code. It is what is inherited between generations, and it is double-stranded, and it consists of nucleotides. And for each individual gene, every person would have two copies of that gene, one of which they inherited from their mother and one of which they inherited from their father. Now, it is important to note that from the time point of the creation of an fertilization of an embryo, when the sperm and egg meet, there are mutations that occur after that. So in many cases, particularly in the case of individuals with mutations in SATB2, the mutation is what we call de novo, meaning that it's something that occurred in the germ cell or later. So it's not present in the parent. In any case, that's a subject for a different webinar. Getting back to the DNA and RNA, so in order to make a protein, the information in the DNA is first transcribed into an RNA. So this RNA is now a single-stranded set of nucleotides that is complementary. It is a copy of a particular gene. So it will only be referred to a portion of the DNA. Now that single-stranded mRNA will then be translated, and that is where the information in mRNA is translated into a protein or a polypeptide. So this polypeptide is a sequence of amino acids, and this is the protein that will be important to the function of the cell. So we're going to look a little bit more at this process, this mRNA to protein process. And at the end of this webinar, I'm going to talk about how different mutations, which of course the mutations would be in the DNA, how they affect the protein. But so what we need to keep in mind is that when that mutation is in the DNA, when the mRNA makes a copy, that mRNA carries that mutation with it. And so the mutation would then be in the mRNA, and that would then contribute to alterations potentially to the protein. So this mRNA is really important as the mediator of this information in the DNA that gets translated into proteins. So this mRNA has a sequence and a few important things about the mRNA. So as I mentioned before, it consists of a sequence of nucleotides, and those nucleotides are represented here by the different letters. So there are 4 different nucleotides. So you can see represented by U, G, A, and C. And those are also color-coded here. So there would be a sequence of these nucleotides. And further, the sequence has a directionality. So what you see here is this, what is a 5' and 3'. And for the purposes of this webinar, that's not so important. The main thing is to know that there is a direction in which the mRNA is read, and this 5' is the beginning, 3' is the end. Another key element about how this mRNA is read is that it is read in groups of 3. So 3 nucleotides will generate what is called a codon, and it might be useful to think of this codon as a word, and that word, in this case the AUG, has a meaning, and that is read as methionine. So each of these 3 nucleotide codons corresponds to a particular amino acid. Another key element of this is that specific codons can refer to either the beginning of a protein, so that's what we call the start codon. So there's a specific codon that will initiate the protein, the translation. And then there's a set of specific codons that are stop codons that will say this is the end of the protein, the polypeptide, and that is shown here. So as you can imagine, there are a number of different possibilities of these codons, and that is what we refer to as the genetic code, and so that's shown here. And since there are many more possibilities of 3 letter codons than there are amino acids, you note that some that the amino acids have multiple codons. So for example, alanine here, there are 4 possibilities, and basically GC followed by any letter will give you an alanine. So this is how that mRNA is read. Another important aspect, and this is the final piece of the mRNA, is that the mRNA has what is referred to as a reading frame. So since the codons occur in sets of 3, there are 3 possible reading frames, and that's shown here. So in each of these panels, it's the same sequence, 5' to 3' is shown in each of these with the UCA at the 5' end. So one possibility is that the reading frame begins at the first nucleotide here, this UCA, that would code for a serine, and then you see that the frame would follow and you get a certain code. The next possibility is that the reading frame begins with the C at the second nucleotide, and then that gives you an entirely different set of amino acids, so an entirely different protein. And finally, in this case, as we see the reading frame beginning with this AUG, we get this particular set of amino acids that we saw in the previous slide. So it's really important that the ribosome is able to identify the appropriate reading frame, because as you can see, that could entirely change the protein sequence. And that's where we get back to this concept of the start codon. So this AUG is where the production of the protein will initiate. So where the ribosome identifies that start codon, that's going to be the reading frame that is chosen. So it's not going to choose either of these other 2 because they do not begin with AUG. The ribosome will continue moving until it finds that, and that will initiate the production of the protein. And so that is shown here. So we have this mRNA strand, and the ribosome is shown here in orange. So in this first panel, it's not completely shown, but it's still there. And it has found this AUG. And then the protein will be produced when the ribosome finds the AUG, and then a different molecule called a tRNA, transfer RNA, that has a sequence that is complementary to that codon will come in and bind, and each of these tRNAs is associated with a specific amino acid. So this one that identifies AUG is associated with methionine, that's the start codon. So as that tRNA will first bind, the ribosome will continue to move 5' to 3', and then the next tRNA will come in that is complementary to the next codon, and it will bring its associated amino acid. And then these amino acids will be joined into a polypeptide chain. And so as the ribosomes move, we will continue to add amino acids, and then the tRNAs on one end will be coming in and on the other end moving out. So that in the end, we finish with our appropriate set of amino acids in a single polypeptide chain. So now, giving that information, we can think about how a different type of mutation may affect the protein. So what's being shown in this image is for all 5 of these rows, again, it's the same, more or less the same RNA sequence. And the top panel is what we would see in a healthy individual, and this is just sort of an arbitrary sequence for an example. But you can see that each of these codons here is corresponding to a particular amino acid. So we have a start codon, which gives us methionine, and then we have 6 additional amino acids, and then we get to this UGA, which is a stop codon. So we have this protein produced, which is 7 amino acids. Now, one type of mutation, and these first, several of these are what we call point mutations. So it's just a change in one single base nucleotide. So this A has been changed to a C. But in this case, the codon, even though the codon is different, they both GGA and GGC code for glycine. And so this is what we call a silent mutation, because it does not affect the protein. So even though the DNA is different and the mRNA is different, the protein that is produced is the same. So that's a silent mutation. In contrast, you could have a mutation where this the second G in this glycine is now changed to a C. And so instead of GGA, you have GCA, and instead of glycine, you get alanine. This is a mutation that is called, referred to as missense, because the meaning of that codon has been changed, and we now produce a protein that has one amino acid that is different than what it should be. And this is a quite common mutation in SAPB2. Another possibility is what we call a frame shift. And here, the example is that a nucleotide has been inserted. So this frame shift can occur with an insertion. So we have the addition of a single nucleotide. It can also happen with the deletion, so the removal of one single nucleotide. And then what happens is that either way, that will shift the reading frame. So these first 4 amino acids are as they should be, but now we have this insertion. And so instead of an AAU here, we get the GAA. And that gives us an entirely different amino acid. And not only is it just this one, but the whole reading frame now has been shifted. So everything downstream is affected. And so that's what you see here with all of these red amino acids that are incorrect. So this would occur in the case of a small insertion or deletion. There are also deletions in SAPB2 patients where you're just missing, you might be missing this entire set of information, and then you get no protein. So that can happen as well. Finally, the last type of mutation that is described here is what we call a nonsense. And in this case, we see that this G has been changed to a U. And so instead of GGA, we now get UGA, and this is a stop codon. So in this case, so it's nonsense because now this particular amino acid no longer has its meaning for coding for a particular amino acid, it's instead a stop codon. And so now the ribosome will stop, and instead of producing the rest of the protein, we're going to get this truncated protein. So all 3 of these types, these last 3 types of mutations are common. in SAS patients. And you might imagine that they would have a different effect, so that this type of mutation where you have only a single amino acid changed versus this one where you might be missing many amino acids, you might anticipate that they would have different effects. But up to now, all the evidence points to the fact that all of these mutations cause disease by a mechanism that is referred to as haploinsufficiency. And so that is sort of pictured here. So on the left, it says homozygous or wild type. You can think of this as a healthy patient. So often we say wild type healthy, which has two unmutated copies of the gene. So there are two copies from two separate chromosomes are both lacking mutations, and they both produce a normal mRNA, and that produces a functional protein. In contrast, this individual that we call heterozygote has a mutation on one copy of the gene. So one copy is fine. It produces a normal RNA and a functional protein. The other copy produces this mutated mRNA, which then produces a protein which is non-functional. And for whatever reason, the evidence points to the fact that all of the mutations in SAPB2 produce a protein that is non-functional. And so what happens is that in these individuals, the heterozygous individuals, which are the SAS patients, the haploinsufficiency is because they simply do not have enough protein to perform the normal function in a particular cell. So we're still in the lab testing whether or not this is true. One of the main questions we have is it in fact the case that these some of these mutations might, rather than being non-functional, they're either partially functional, or it's possible that they are doing something inappropriate, which might be making a certain process function worse than if it were absent. So these are questions that are currently being investigated in the lab, and I hope that someday soon we'll have answers to that question. So that is all for this webinar.